Johnny Kirti Salik and I, Joe, will be presenting on women's contributions to vaccines. We will be highlighting women who made direct contributions to the development of vaccines, as well as women who made notable contributions.
Both Pearl Kendrick and Grace Eldering developed the pertussis vaccine after a virulent strain infected a nearby city's population of children. In addition to this, they pioneered the field of clinical trials by designing and directing the first large-scale controlled clinical trial for the pertussis vaccine.
Before Isabel Morgan, it was accepted among virologists that vaccines could only be effective or functional by using a live attenuated form of the virus. Morgan and her team spearheaded the development of inactivated vaccines by being the first to inoculate monkeys with an inactivated form of the poliovirus, proving that inactivated vaccines can provide the same long-term immunity as live attenuated vaccines.
Rachel Schneerson worked to improve the immunogenicity, which is the ability of foreign substance, such as an antigen, to provoke an immune response in the body of a human or other animal, of the Haemophilus influenza B vaccine, or HIB vaccine. In addition, she started the development of conjugate vaccines for other bacterial pathogens whose surface polysaccharides could serve as a protective antigen. Kizzmekia Corbett is a senior research fellow at the Vaccine Research Center at the National Institute of Allergy and Infectious Diseases and chief of the Viral Pathogenesis Lab. Through the contributions of her and her team, they were able to produce an effective vaccine for the biggest pandemic in a century in an expeditious amount of time.
Pearl Kendrick received her degree at Columbia University in bacteriology and was recruited in 1914 for a new branch of the Michigan Department of Health in Grand Rapids. Grace Eldering was Pearl's partner at the Michigan Bureau of Laboratories, and she was brought into the branch in 1932. Due to budget cuts, talented women were sought after as they were cheaper to pay, but because of Pearl, the new branch got the opportunity to prosper.
Later in 1932, there was a whooping cough outbreak in Grand Rapids. In response to the outbreak, Eldering and Kendrick's lab developed and improved methods for growing the pertussis bacillus. They modified the standard growth medium to have more rapid and profuse growth of the B. pertussis colonies, which in turn allowed for them to determine when a child was infectious to peers, dictating the quarantine length. By inactivating it and creating a safe vaccine, they have conducted the first successful large-scale controlled clinical trial of the pertussis vaccine. And in 2019, they were memorialized with a statue in Grand Rapids, along with lab technician Linnie Clinton Gordon.
Isabel Morgan received her PhD in bacteriology at the University of Pennsylvania in 1936. She then worked at the Rockefeller Institute and ended up at Johns Hopkins in 1944. Isabel Morgan and her team worked hard to disprove the popular notion held prior that vaccines had to be live attenuated to work. Morgan researched the effectiveness of inactivated vaccines, specifically regarding that of the poliovirus. After 5 years of research, Morgan and her team were able to successfully inoculate monkeys with an inactive form of polio, demonstrating that her vaccine was effective against 2 out of the 3 strains of polio. This was done 5 years before Jonas Salk created his vaccine. She quit polio research in 1949 before actually creating a vaccine for humans, and this was likely due to a few reasons. In interviews, she explained her fear for the next step of vaccine testing on human children, and because this was around the time that World War II had ended, Morgan became focused on becoming a homemaker for her husband and son. Had she stayed her course and continued the development of her vaccine, she would have likely beaten Salk in the race and would have the namesake over the polio vaccine today.
During the time that Rachel Schneerson was conducting research, Haemophilus influenzae B, or HIB, was the leading cause of meningitis among children under 5. Even with effective antibiotic treatment, still 5% died and 30% were left with an intellectual disability. At the time, polysaccharide vaccines were an effective treatment for HIB. However, younger children, who were the main group that was affected by HIB, had immune systems that did not respond as well to polysaccharide vaccines as compared to adults and older children because their immune systems were not as developed. Schneerson and Dr. John Robbins worked to develop a conjugate vaccine that connected the weak polysaccharide to a protein that can be easily identified by an infant's immune system, making it much more effective for infants and younger children. Today, it has become a part of routine immunizations given to children, and because of this, it has led to a 99% decrease in HIV.
Corbett was a senior research fellow for the Vaccine Research Center at the National Institute of Allergy and Infectious Diseases. She was the chief of the viral pathogenesis lab during COVID, and she is known for leading the team that created a highly effective, easily manufactured vaccine against COVID-19, which was delivered and authorized for use in under a year. Along with her research activities, Dr. Corbett is an avid advocate of STEM education and vaccine awareness in the community. Before COVID, Dr. Corbett was researching the coronavirus category of infections for 5 years, and her work on the spike proteins proved influential in the creation of her most famous work, mRNA 1273, made specifically to combat COVID-19. It took an unprecedented 66 days to go from viral sequence release to FDA-approved phase 1 clinical trial, which was one of the fastest timelines in vaccine production.
The women we have described today in these slides are just a few of the many women who have had an immense impact in the world of vaccine development and research. But there are very few who are awarded the proper recognition that they are warranted. These are some of the most important women who we feel have made the biggest contributions that go unheard, but many unsung heroes merit the same, if not more, commendation and acclaim. Thank you.